Yeah, hello everyone. I'm delighted to introduce Dr. Johan Skog, our keynote speaker. For those who don't know, Dr. Johan Skog, he obtained his PhD in gene therapy and virology and a master in biomedicine from Umeå University in Sweden. Then he joined as a postdoctoral fellow at the laboratory of Dr. Xandra Breckfield at the Harvard Medical School to study the role of extracellular vesicles in glioblastoma. And he has been the first to demonstrate the transfer of messenger RNA from extracellular vesicles to recipient cells, which resulted in a groundbreaking publication in Nature Cell Biology in 2008. His scientific work generated more than 20,000 citations and still serves today as a framework for a current understanding of the role of EVs in intercellular communication. In parallel, he developed interest in using extracellular vesicles as disease biomarkers, and in 2008, he co-founded the company Exosome Diagnostics, now a brand of Biotechny, to develop a EV-based diagnostic test for human diseases. And noteworthy, in 2016, Exosome Diagnostics launched the ExoDX Prostate test, which is the first clinical-grade exosome-based urine test to assess the risk of developing prostate cancer. Dr. Skog holds more than 100 patents in the EV field and pursues his efforts in the development of EV-based diagnostic tests. And as such, his group recently published in 2021 preliminary data of a second exosome-based urine test to assess kidney transplant rejections. So Dr. Skog's achievements illustrate the strong potential of extracellular vesicles as biomarkers and the feasibility to translate discoveries from bench to the clinic. So Johan, thanks a lot for speaking at the Urinary EV Symposium today, and the floor is yours.
Thank you so much, Fabrice. Can you hear me okay, first of all?
Absolutely.
Great. Okay. Yeah, thank you so much for allowing me to present here today. I'm excited. This is it's been a great meeting and a lot of interesting talks. So I will talk a little bit about exosomes, from exosome-based biomarkers from bench to bedside, and talk a little bit about our experience and go into some of the details of the urine EV tests that we have launched. So there's been, of course, a very rapid growth of exosome-based science, and basically the interest has spurred a lot of developments in diagnostics as well as therapeutic applications. And I think very few new areas like the exosome field has sort of moved as quickly as this, where we saw the world's first exosome-based test come to market less than 10 years after the foundational RNA discovery. But I think another important factor is that these are really creating waves in the field of research just for the functional aspects of exosomes, where they're also part of this intercellular communication. I can transfer molecules from one cell to the other, and enables transfer of molecules that are not normally secreted. So that was sort of a really big deal. Another aspect of the exosome component that has been discussed a lot at this meeting is, I think we have to move forward with a multi-omics approach, because when we're trying to look at complex diseases, especially complex diseases like cancers, looking at single omics, looking at a protein or just a cell-free tumor DNA molecule or so, will not give you sort of a complete enough picture of these complex diseases, where if you have only a single omics here, you're only describing one aspect of the biology of the disease. So the elephant in the room here is, of course, that you need to utilize and combine this multi-omic approach to get a proper description of the diseases. And we feel like the exosomes are perfectly suited for this because of this package of multi-omics that are in there. So it enables you to to look at EVs, of course, with the content of the proteins, the lipids, the RNA and the DNA that's associated with vesicles, but combine that also with other liquid biopsy entities like ctDNA or proteins. And I think this is needed to provide a more comprehensive understanding of the molecular changes that are actually happening in a variety of different diseases. So we use exosomes as a liquid biopsy. And typically when you hear the word liquid biopsy, you're thinking of there are three molecular entities that are typically discussed. And those are cell-free tumor DNA, exosomes, and circulating tumor cells. And I think another aspect of this multi-analyte approach is that you're actually surveying very different types of biology when you're looking at these different types of entities, where ctDNA, for example, has moved very quickly in the liquid biopsy space. However, it's released from the dying part of the tumor. So that cells have died from apoptosis or necrosis, but it enables you to do mutation analysis, methylation analysis, and fragmentomics. Whereas the exosomal compartment is an actively released process that can be used also, since the RNA mirrors the DNA, it's an amplifier of the mutations on the DNA. So you can also use that to look at mutations, but more importantly, the RNA transcription pathways that are dysregulated in the disease, and also enables you to use splice variant analysis and fusions in this biomarker space. Another important aspect is the sheer number that is released here from the EVs, that one cell can secrete tens of thousands of these EVs per day, but that same cell doesn't secrete any cell-free tumor DNA until it dies, when it secretes 2 copies. So there's just this vast difference in abundance of the molecules. And to illustrate that abundance, I actually really like this video that Vervey et al. Published on cancer cell that illustrates the abundance of the exosome release, where here you actually see a cancer cell that was modified with a fluorine tied to the CD63 molecule. So every time you see a light flash on this cell, it's release of a multivesicular body to the surroundings. So you actually get release of dozens of exosomes. So this video clearly illustrates sort of the dynamics of the release of these vesicles to the surrounding. So when it comes to the exosome isolation part, there's of course a lot of different preferences for what methods to use. And I think there's been sort of almost a holy grail in this space where people have been looking for a one-size-fits-all solution. We quickly came to the conclusion that that's not where we think the field should go. At least that's not where we wanted to go. We learned that there needed to be a tailored approach depending on the biomarker that you're actually interested in and what the biofluid matrix looks like. Because doing an EV isolation from a plasma sample is very, very different compared to an EV isolation from a cerebrospinal fluid sample or a urine sample. So you have to optimize the protocols depending on what you're trying to do, and also optimize them depending on if you're trying to target an intravesicular RNA molecule or a protein or something on the surface of the vesicles, because there are very different considerations that you have to take into account there. But at Exosome Diagnostics, we utilize our Exsolution series of isolation products for any of our clinical programs. And we pick the method depending on what we want to achieve. So in cancer, there's often a need to look at mutations, for example. And we know that mutations exist on both cell-free DNA and exosomal RNA. So we often utilize our Exsolution Plus kit for that, because this co-isolates the mutations on the RNA, on the exosomal RNA and the cell-free DNA. So we actually get a higher sensitivity assay for those liquid biopsies. And our UPrep is our urinary exosome isolation method whenever we're doing prostate cancer or bladder cancer or anything like that. And these are just representative pictures from the lab and how we do this on a daily basis in a high throughput manner, where it's the most important thing when it comes to the isolation of the exosomes is maybe not the sort of the same as the purest view of EV isolation. The most important thing from a biomarker aspect is that you are getting your biomarker out reproducibly every time. And there's sort of less emphasis on sort of the actual method to get there. You just have to ensure that you can do it the same way every time, and that the biomarker that you identified is reproducibly isolated. I know that in this exosome field, often when we go to meetings, there's this notion in the liquid biopsy space that EVs are complex to isolate, and that you cannot do this reproducibly in a clinical setting. But that is, of course, not true. And I like to illustrate that with this reproducibility slide, where we didn't design this experiment as a true reproducibility experiment. Instead, I actually called up our CLIA lab, our clinical laboratory lab, that are processing patient samples every day. In every batch of urine samples that they isolate the exosomes from, we include a control urine sample in every one of them. And so I asked the lab to actually give me the data from the last 440 replicate extractions. So this corresponds to dozens of operators, thousands of clinical samples over years. And as you can see here, the qPCR CT values across the batches, and if you zoom in on batch 5 in detail here, you can see that the reproducibility of the exosome isolation, the RNA isolation, the reverse transcription, and the qPCR has a standard deviation of about 0.4 CTs, which is similar to what you expect from a standard qRT-PCR experiment. So the exosomal isolation part can be done incredibly reproducible. So if you optimize those processes, it opens up this huge field of diagnostic applications, because we can harvest these exosomes from a variety of different biofluids. So you can do that from urine, you can do that from plasma, you can do it from saliva, cerebrospinal fluid, etc. Most importantly, you need a sort of a clinically applicable exosome isolation platform that can isolate the biomarker of choice. And that's really, really important. I think we often get questions like, well, we isolated in our biomarker discovery, we isolated the exosomes with this complex differential centrifugation protocol with density centrifugation, etc. And now we want to move it into the clinic. And those biomarkers may not always translate because the methods are different. So we think that it's very important to also do the biomarker discovery using the approaches that can be used in a clinical workflow. And so if you do the exosome isolation properly, we have mostly focused on the RNA in exosomes. And if you do the extraction properly, you actually get really good quality RNA out of these exosomes. That enables you to do global RNA transcriptome profiling. And we have mostly focused on the long RNAs. I know in the literature, there's maybe a little bit of bias towards microRNA or small RNA profiling. However, we have found that if you actually do the isolation in a way that minimizes the RNA, partial RNA degradation during the isolation process, you actually get far more material out and more diversity of RNAs on the long RNAs. So it enables you to do things that you can do pathway mappings, for example, to understand if the MAP kinase or the EGFR pathway is upregulated in the patient population, or to understand those biological properties. And that's important not only for biomarker research, but also for therapeutic development. When you have a new therapeutic in the clinic that you want to evaluate in a clinical trial, and you're not actually sure if you're hitting the right pathway in vivo with the drug. So having this as an opportunity is very important. And when you have the RNA out from these biofluid samples, you can do the whole transcriptome, or you can do a more targeted approach with an exome sequencing, exosome plus link RNA, or even a PCR-based readout of this to do the analysis. And what we actually see is that from 0.5 ml of plasma or 10 mils of urine, you get the same diversity of RNA as you get from a tissue sample. So about 30,000 of the 56,000 genes in GenCode and about 17,000 of the 20,000 mRNAs in GenCode can be reliably detected with these methods at this sequencing depth. You can always, of course, get a little bit more by sequencing deeper, but we did a comparative study with tissue at the same sequencing depth. And in every patient sample that we analyze with this method, we spike in 92 exogenous control RNAs so that we can, so these 92 control RNAs are of different sizes, different lengths of RNA, and at different molar concentrations. So in every readout, we can ensure that the RNA that went into the library, we have a high sensitivity, good LOD, and a wide dynamic range of the readout of the RNA. Here's an example of that and how we assess this in every patient sample that we run, where we actually plot the expected copy numbers of the spike-in controls versus what we see in the actual sample. And you can see that it's extremely robust, where we can see down to 10 molecules of RNA with a large dynamic range spanning over 5 orders of magnitude. With PCR-based methods, you can go down in the stochastic range as well. But for global transcriptome profiling, I think this is extremely useful. It's also important that you can do the downstream analytical processes in a high reproducibly manner. So here, this is an example. When you're doing RNA sequencing, when you're testing the reproducibility, you have to test different batches, not only of your sample, but batches of reagents that you're getting from your manufacturer, because sometimes they like to actually try to improve on the library prep reagents. However, an improvement would actually be detrimental to a diagnostic because you're changing something. So this just illustrates that you can actually do it with an optimized process where the correlation is as close to 1 as you can imagine. This is based on 24 replicates of the same biological samples run in 3 different batches. And what was interesting when we tested all types of different biofluids here, but urine libraries shows the highest proportions of transcripts with more than 80% coverage of the RNA transcripts that are over 1 kb in length, which translates to the fact that urine RNA, if you do it correctly, has an incredibly high quality and really good size distribution of RNAs in urine EVs. And that that has very big implications for any diseases in the urogenital tract. So one example that you can look at is prostate-specific RNA targets in urine. So in this particular experiment, we applied that whole transcriptome sequencing approach on patients where the urine was collected before radical prostatectomy and then another sample after radical prostatectomy. So these are matched samples from a large cohort of patients. And these were urine samples that's been stored about 10 to 12 years in our freezer. So we have actually outcome data from these patients. We've seen in our stability experiments that we can do this on samples that's been stored actually over 25 years in the freezer. So that's quite remarkable for an RNA, I think, that is otherwise notoriously known to be unstable. But in this experiment, we of course then in the pre versus the post RP samples, we can see the delta, what genes disappears after the prostate is removed. So we see the hundreds and hundreds of genes that are truly prostate specific that disappear after the prostate. To verify that this was really prostate specific, we plotted the top 20 prostate specific genes based on GTEx. And then we looked at what actually happened to those genes in our data set. So we actually have more genes that are prostate specific, but this was actually just a verification. And you can see in the blue, which was the pre-RP samples versus the orange one is the post-RP sample. You can see that they all went down or almost disappeared after the prostate was gone. When we looked at with the same approach, we looked at genes that are known to be expressed in the kidney and known to be expressed in the bladder. Those were not differentially expressed after radical prostatectomy. So that was sort of a verification that that this is truly a prostate-specific approach. And so urine is really a gold mine of targets that you can harvest for a variety of different urogenital tract diseases. But in addition, you can look at fusions and splice variants that are in urine. This is just an example of one of the fusion genes called TMPRSS2 that's expressed in roughly half of prostate cancer patients. And we actually had the urine sample taken pre-biopsy, and then we had a tissue from after radical prostatectomy where we could actually look through the entire organ for TMPRSS2 expression. And there's a remarkable concordance. And we had 3 patients where we couldn't detect it in the urine sample, and 1 patient where we found it in the urine, but not in the tissue sample. So this was surprising. So that we actually went back and we looked at this more in detail. And it turns out that there was actually the first initial tissue analysis did not have access to one of the most anterior parts of the prostate. And it turned out that that was the missing part. There was actually a piece of tumor in the anterior part that was not analyzed in the first one. So this one was a false negative in the tissue, and we picked it up in the urine sample. You can also look at splice variants that are implicated in prostate cancer, like the androgen receptor and the ARV7 splice variant. That can be detected in plasma. There are tests out there that are now utilizing circulating tumor cells, looking at ARV7 in prostate cancer patients. But as you know, the pre-analytical conditions to actually get to the CTCs can sometimes be cumbersome. And having something that is more stable, where you can utilize a standard plasma sample that can be frozen, has a lot of benefits. So moving into sort of the clinical arena, I mentioned from a urine sample, you can do global profiling. You can actually look at well over 30,000 genes and over 100,000 different targets if you're including splice variants and others. However, in this clinical setting, we developed a prostate cancer test where you only need to look at 3 target genes. So it can be done by a very simple qPCR analysis. And as Fabrice mentioned, we've also sort of tagged on to this urine sample collection matrix for a kidney transplantation assay that we have now licensed out to Thermo Fisher. But these are all based on a urine collection kit that can be done either at the clinic or at home. The patient can just urinate into a cup and send it in for analysis. So when you're looking at a urine exosome collection, a urine sample will carry exosomes from the kidney, from the bladder, and the prostate, if it's a male. And we don't see a whole lot of contribution from the peripheral targets. So whenever we want to look at peripheral disease, we prefer to look at plasma or other biofluids that are more representative of systemic approaches. However, it's really a gold mine for the urogenital tract. And we test the different urine fractions to understand where these really are. So there's this first catch urine collection device where you urinate into the funnel. The first 20 milliliter or 10 or 20 milliliters of urine ends up in this compartment here. The green thing is a float. So as the first catch urine has been collected, the float goes up, shuts off the urine collection, and the urine goes out this way into the toilet. So we taped 5 of these together. So we basically got all the different fractions of urine, and we provided them to individuals. And then we looked at the biomarker distribution in the urine fractions, and we could clearly see that the prostate fraction is really in the first catch. And then in the midstream and last urine is mostly the stable flow of the bladder markers. So the EPI test, or the ExoDx prostate test, is developed as a risk score to stratify your likelihood of finding high-grade prostate cancer on initial or repeat biopsy in men 50 years and older with a PSA of 2 to 10 nanogram per milliliter. You may ask, well, why is it so specific for that population? Well, it was developed for this population for a very clear reason. We wanted to develop a biomarker in the space where it's really clinically needed. If you have a PSA of 50 or 100, you're likely to get a biopsy because PSA works really well when the PSA is very, very high. However, PSA does not have very good discriminatory performance in the gray zone of PSA from 2 to 10. So that's really where we wanted to focus to add additional clinical value to the clinician. We know that our test performs equally well with a patient of a PSA to 20. However, the clinical utility at those upper PSA levels is sort of debatable. So we developed this at-home urine collection or in-office collection where the patient can just pee in a cup, send the sample in. It's very stable, stable at +4 for up to 2 weeks for these biomarkers, or in the freezer almost indefinitely. We look at 3 genes. So these are PCA3, SPDF, and ERB that are well known in the prostate cancer space. They go into an algorithm where it generates a single score from 0 to 100. And the higher the score is, the higher the likelihood is of finding high-grade prostate cancer upon a biopsy. So what's unique about this is that it gives you completely independent molecular information. In this test, we chose deliberately not to include other standard of care parameters in the score. We know that it's been popular to maybe add the value of PSA or add other callicrines as well as ethnicity, age into your algorithm to improve the performance of the test. However, we wanted to stay away from that because that's information that the clinician already has. If you build it into the score, the clinician would actually need to stop using those standard of care factors in their decision making, which they won't do. They will always take use, if they think that they have a higher risk patient, they will weigh that into their decision matrix. So this is completely independent that they can add to the standard of care factors. The way this test has been developed over the years, it used to be called Exosome 106, sort of in the development phase of this test. And there's a lot of work that goes into developing a test. As you can see in the early days, we did 4 clinical cohorts that actually we haven't published on. And that was where we generated our signature. So we didn't publish when we had the signature validated. What we wanted to publish as a validation paper was a specific cut point. So at the signature validation step, we found the most optimal cut point of the score where the clinical decision making would happen. We actually didn't decide that cut point ourselves, but we had invited over a dozen key opinion leaders in the prostate cancer space that sat down and had basically a conference looking at our validation data, and they set where they thought the cut point should be. And that turned out to be at 15.6, because they wanted to have a test that had a really good rule out capability, where you have a cut point that rules out the chance of high-grade prostate cancer, so you can safely forgo a biopsy. That's where the clinical decision making would be. So after they had set the cut point, we published our validation study, and then we've repeated the validation study as well, because that was needed for insurance and payers, as well as guideline inclusions. We performed a clinical utility study, which is actually a unique study showing how the utilization of this test benefits patients. So after those validations, so we of course have to we continue to validate the performance of the test in a variety of different applications. So we have actually not only 2, but actually 3 independent prospective validation studies of the test, as well as validation in patients that had a prior negative biopsy. And in addition, one of the issues with prostate cancer is the biopsy itself, where it's known that the biopsy, or 12-core trust biopsy that's usually performed, has a very high chance of missing cancer and missing high-grade prostate cancer. So we did also an analysis to look at patients that were confirmed to have a certain diagnosis after radical prostatectomy. So that test performance at that validated cut point of 15.6, you want to have a high sensitivity and high negative predictive value. So they were all over 90%. And if you define high-grade prostate cancer as more like Gleason 4+3 and higher, or ISIP3 and higher, the NPV actually goes up to 97%. So that's the cut point analysis. However, we found that this score, the exosome prostate test is not really it's similar to most other biomarkers, that it's a continuum, that the higher your score, the higher your likely, or the higher the biomarker, the higher the likelihood of prostate cancer. So when you start to plot sort of bin the patients, you can see that the higher the score, the higher the chance of finding high-grade prostate cancer upon a biopsy. So at a score of 30, you have a 30% chance of finding high-grade prostate cancer. At a score of 50, you have a 50% chance of finding high-grade prostate cancer. You may ask, what's this? Why does it drop off here? Why does it not continue up to 100? Well, you know, that's the caveat of doing biomarker studies where the gold standard is not perfect. The gold standard in a biomarker study here is 12-core trust biopsy. And the PROMISE trial showed that 12-core trust biopsies misses 50% of high-grade prostate cancer. So that makes it impossible to actually carry on up to 100 with this one. So if you see someone that has a biomarker for prostate cancer that is near perfect with an AUC well over 0.85, you know it must be overfitted, because you cannot achieve that with a flawed gold standard where the biopsy misses 50% of high-grade prostate cancers. So how are these this test now done or used in the clinic? So it's mostly when you have a patient coming into the clinic. If you have 3 patients coming in, they have the same age, similar PSA levels, and all other standard of care factors are very similar. Who will get a biopsy here? Well, in this scenario, it really depends on the patient, and it depends, the patient's willing to go through a biopsy, and it depends on the doctor that the patient is seeing. And there's nothing here with the standard of care factors that would guide you to biopsy one patient or the other. However, the EPI test can actually give you completely independent information based on the molecular signature in the urine that identifies now patient B and C as higher risk individuals and patient A as a low risk individual. And that's actually what turned out to happen after the biopsy. And this in our clinical utility study, we actually found that urologists that had access to the EPI score, they had found 30% more high-grade prostate cancer compared to a blinded control arm that just used standard of care. So this clinical utility study was a prospective randomized prostate cancer biomarker utility study. So utility study is different. It's not the validation study, because the validation has already been done. This is where we want to understand how the biomarker improves the outcome of these patients. So for this reason, we had that urine collection, and every patient gets an EPI test. However, after the urine has been collected from these patients, they were subject to randomization. So they either went into the EPI arm, where they actually would receive the score back, or the control arm, where they did not receive the score back. And the doctor had to, together with the patient, decide what to do. So they proceeded with standard of care. They can use any other factor in their decision-making process to go through with a biopsy or not. Whereas here they use standard of care plus the information of the EPI result. So this was over 1,000 patients across 24 different centers and 72 different physicians. And what they found was that in the EPI arm, the doctors that have, or doctors and patients that had access to their EPI results, they had a reduction in biopsies when the EPI test was negative. And the EPI negative patients were not identified by other standard of care factors. And so they received more biopsies in the blinded control arm. Because remember, we had access to the EPI score from the control arm, but they didn't receive them. So we could actually cross-check them after the study was done. And we found that the EPI negative patients in the control arm received far too many biopsies. However, among the EPI positive patients, that led to finding these 30% more high-grade prostate cancer compared to the control arm due to the better compliance, because the doctor and the patient knew that they were at a higher risk and went through with the biopsy. So I mentioned sometimes the biopsy is wrong, right? You do a biopsy and you miss the lesion. And this is due to the fact that when you're doing a 12-core truss biopsy, you're only sampling a very small part of the prostate. And there's a risk that you're actually missing the lesion. And since it's also a multifocal disease, you can actually miss the high-grade lesion. So what we wanted to understand here was that, so we followed about 2,000 patients that had an EPI result done before the initial biopsy. And out of those 2,000 men, over 300 men proceeded with a radical prostatectomy due to the finding of the biopsy. However, when we looked in detail, 111 of those men, they received a radical prostatectomy despite the fact that they just had a grade group 1 or Gleason 6 at the biopsy. So these men are definitely candidates for something called active surveillance, where they're just being actively monitored instead of going for full-blown treatment. But it's a personal choice, and some men still decides to go for radical prostatectomy, even though they have a Gleason 6 cancer. And it turns out that in this cohort, roughly half of the men that had a Gleason 6 on a biopsy were upgraded to a grade group 2 or higher after the radical prostatectomy. And I think that is also one of the many reasons that some Gleason 6 patients, they continue to get repeat biopsies to ensure that the first biopsy was not wrong. But remember, when the biopsy is wrong in the initial biopsy setting, in our validation trial, they're actually called as false positives by definition, even if it's wrong. So here we actually wanted to understand what happened with those false positives. Were they really, were there any indication that they actually had cancer in the first place, or higher grade cancer in the first place? And it turned out that when we started to stratify these patients, that only an EPI result that was done pre-first biopsy, the ones that were upgraded to a grade group 3 and higher, they had a significantly higher EPI score, actually twice as high EPI score as patients that were confirmed to be lower grade cancers. And when we plotted the same patients, we looked at the PSA values, we looked at the PCPT risk calculator that includes PSA, ethnicity, age, etc., all the clinical factors in an optimized algorithm, or the European risk calculator. There was actually no sign that these men were going to be upgraded at radical prostatectomy. So there was nothing that the doctor could see that these were actually missed on the first biopsy. Only EPI had identified those men already in the pre-biopsy setting. And this is the ability to detect higher than GG2 patients in that population. And when we looked at the pre-biopsy EPI score, they nicely correlated with the post-RP pathology results at the validated cut point. And there was no GG1 cases with an EPI score below 15.6 that got upgraded to GG2 and higher. Whereas when the EPI score was positive, there was a significant amount of patients that were upgraded to GG3 and higher. So to summarize, this exosome, urine exosome test is a completely non-invasive urine test. It does not require digital rectal exam or anything like that, and prostate massage. And it really helps you inform the risk of high-grade prostate cancer on a biopsy. And it identifies the right men to biopsy, right? So any urologist that have access to the score finds 30% more high-grade prostate cancers than the doctors in the same clinic that were blinded to the result. And it avoids a biopsy in epi-negative men. And when we looked at our utility trial data, we saw that those epi-negative men, they were proven to be low risk for high-grade prostate cancer, but they received many more unnecessary biopsies when the doctor did not have access to the result. And it gives you actionable information that cannot be found by other standard of care factors used in the real-world urology practices. And it was the only parameter that was associated with high-risk pathology after radical prostatectomy in men with Gleason 6 or grade group 1 on the biopsy. So we really think that this liquid biopsy may address some of the limitations of the biopsy heterogeneity. And we think that there's a very clear case for utilizing this type of analysis in patients on active surveillance. However, we're doing further ongoing studies in this population. And this test has been included in the NCCN guidelines for early prostate cancer detection since 2019. So it's helping patients every day. And with that, I say thank you for listening, and I'll take any question.
Thank you, Johan. That was a very inspiring talk, very educational, and also impressive amount of work over the last 10 years. I'm going to just go through a few questions before leaving the mic to Uta and Jane for the panel discussion. So I'm just going to rephrase a couple of questions to go faster, but a question regarding the batch, the batch-to-batch reproducibility that you demonstrated at the beginning. So there was a question, very simple question. Does each batch consist of multiple urine samples that were pooled, or it was like really from individual samples that were measured?
The batch urine samples are from pooled urines because we need to do an enormous amount of extractions. So these are pooled urine samples. We've done smaller experiments. If you only do, for example, a dozen replicate this was actually representative of thousands of patient samples, right? Whereas in individual, you can do maybe a 24-hour urine collection and do maybe a dozen replicates. And if you do that, it's the same. The single urine samples behave in the same way as a pooled urine sample.
And so there is a couple talks about the variability, like daily variability in urine, urine content in extracellular vesicles and different pre-analytical parameters. To minimize the batch-to-batch variability in your like clinical grade urine test, does it include also well-defined urine collection and processing protocols? And I know that you guys collect the first 20 ml, but is there any restriction to make sure that there is not much variability between the batches?
Yeah, I mean, that's the disadvantage of urine. The advantage are clear. I mean, that's all that I talked about. The disadvantage of urine is that it's not a homeostatic liquid in that sense, right? It depends on the patient's hydration level and when did they urinate the last time, etc. So we are, that's why it's important that you verify your biomarker appropriately and you have the right normalizer in your setting to avoid some of the issues with the prostate specific markers is that we don't want the patient to have just urinated. right? If you can imagine, if they come to the clinic and they go to the bathroom and they pee, and then they come into the doctor, and then you want to collect a urine sample that supposedly should have prostate exosomes in them. The patient have now washed out those prostate exosomes in the first urination. So we require the patient to wait 1 hour. And that 1 hour is sufficient to regenerate some of the prostatic secretions that end up in the urethra to be washed out. So that's the condition that we use.
Very good. So I'm going to ask a last question. There was a good discussion. Something that to keep in mind for everyone, Maya asked a very good question about the implementation of multi-omics to better understand the biology and see how it can be translated to the clinic. I think that's a good question for the panel discussion. So just keep that in mind. But there was an interesting question from Marvin Drost, and also Elena was discussing about, have you looked at the clinical utility of the EP test in combination with MRI, knowing the performance of MRI today to avoid biopsy, unnecessary biopsy in men with like grade group 1 or even BPH?
I have. Of course. Let me see if am I sharing the slides?
Yeah.
So yes, absolutely. MRI.
We're ready for the answer.
Yeah, if you're talking about MRI, it has a very high utilization in Europe, and it's rapidly increasing in the United States as well. MRI is also a useful tool, of course. However, it suffers from a lot of verification bias, where doctors see something on the scan and they go in and they biopsy that and they find something. If they don't see something on the scan, they don't target that region. And we know that MRI actually misses a lot of high-grade prostate cancers in that setting. So you need to have a very controlled study where the biopsies are done also in the MRI negative patients. I think the PROMISE trial is one of the gold standards that sort of defines the performance of MRI in that settings, because they did a 96-core biopsy as well as a 12-core biopsy of each patient. But what we found in our studies is that these molecular biomarkers has an additive value to the MRI and can actually help limit some of the deficiencies of those imaging-based biomarkers, because you get molecular content that adds value to it. We don't want our molecular biomarkers to be perfectly correlated to MRI. They need to be correlated, but not perfectly, because if otherwise there's no synergistic value of them. And when we actually look at PIRADS versus the EPI score, they are nicely correlated, as you can see. And look at the proportions of high EPIs in PIRADS 5s, 4s, 3s, and 2s, etc. So it's nicely correlated, but it's not perfect. So it provides additional synergistic information in the evaluation of these patients. And we haven't published that, but we're doing an MRI study that will give more guidance on the exact utilization of it. But one example that was published previously by Decal et al. Is how you can utilize MRI with molecular information and get actually better decision outcomes.
Thanks a lot, Johan. I'm going to let Jan and Uta to continue with the panel discussion that you're actually participating to. And if you have a chance to take a look at the chat, there's a couple of questions that I think deserve to be addressed. Thank you very much, Johan, for your talk. It was amazing. Always nice to have you here, and through your work, inspire us to look for better biomarkers. So thanks again.
Thank you.